So here we are again. I think it might work. Yes, I can hear myself, and I would like to welcome you again to another Saturday Microscopy live stream. Happy that you could join me again. Just give me a second to get everything sorted out. Audio seems to be working. I can also see myself on YouTube. Chat seems to be working. Yeah, I think we're just gonna get started again, and we're gonna take it from there. Okay. Yeah. And you've probably already realized a little bit of new design here. I put myself into this little square with rounded corners. Occasionally, I like experimenting around a little bit as well with the design. Yeah. So, today I want to do something very traditional again. I want to look at a couple of water samples, a couple of algae again. I know that this is something that I've been doing every now and then. Yeah, I got collected today 4 water samples to look at. I did not look at all of them yet. However, I also caught a couple of larvae. I think it might be some kind of mosquito larvae or related in here, 3 of them. Which I would also like to put under the stereo microscope, and we're gonna take it from there. Okay, yeah. First of all, yeah, if you are new to this live stream, please feel free to write comments in the chat. I'll try to answer them as far as I can. And of course, hello all around the world. I'm now not going to go through all of the comments, yeah, because yeah, there are many people who've joined in. So a little bit some organizational stuff first. The specimen that you see right now here is actually a specimen that we also looked at last week. And I'm going to show you quickly. This was the enrichment culture, which we made 2 weeks ago. Where is it? Here we are, right? If you remember, if you joined in also last week, this is a pond water sample, and I dropped in a couple of wheat grains 2 or 3 weeks ago. Basically, this is how the the water sample looks now. It turned much more green, so the algae started to grow. There are now also a whole bunch of rotifers in here. Also, yeah, other planktonic algae also started to increase. So it's a pretty active water sample. What I have done is as the water evaporated, I added more and more water to it to compensate for the evaporation. And this is basically what we've got here. Okay, and there is a couple of days ago, I also uploaded a video about the oxygen requirement of bacteria. It's yeah, just a few days ago. If you're interested, you can also watch that after the live stream, where I specifically looked at the movement of those bacteria in the background. Okay, because they started to form so-called aerotactic bands, and we're going to see if this also works here right now. So those tiny little things here that you see in the background, these are bacteria, and they started to yeah, move towards oxygen, those tiny bacteria in the background. But there are so many algae now all over the place which produce oxygen that maybe they're not going to move into a specific direction. Okay, so one comment here, lots of Senedesmus. Again, if you look, for example, of those algae, 4 of them, that's Senedesmus. We can go up with the magnification. Yeah, so it's I think quite an interesting looking culture. So, and what I would like to do today is to look at a few other water samples as well. Yeah, you can see that there is now some water movement going on here as well. A whole bunch of diatoms are present. You've got several ciliates, you've got rotifers, yeah, lots of stuff is yeah, going on here. Yeah. Yeah, right now I'm using my 60 times objective, you know, so the magnification is fairly high. And yeah, what we're going to do then is we're going to have a look at a few of the other water samples. And another thing I want to show you is a little failed experiment because I tried to make a slide with my hair cross section, which did not quite work out. I'm just going to share it with you later in any case, because yeah, I just wanted to show this to you as well. Okay, look, okay, here we go again. And maybe we're going to look at a slightly lower magnification. Yeah. So it's yeah, it's quite a party going on here. Usually, water samples like this are always best observed live and not yeah, and not with a in a permanent mount, because simply the movement is simply gone. Yeah, see a whole bunch of other larger creatures as well. Let me turn off the arrow. Yeah, so let me, okay, yeah. So it's quite a fascinating thing to observe. Yeah. So this is without centrifugation or without anything. Yeah. So, and what you might notice, and this is something that I tried to demonstrate in my other video that I made a couple of days ago, is that those bacteria in the background, they are going to start to move towards the edge of the cover glass, towards more oxygen. But that is something that we actually might not be able to see well today, because there is simply too much oxygen present anyway, because of so many algae, right? Look at all of this density that we have here. Yeah, it's quite something going on. So the recommendation is if you don't see anything interesting right away, then wait a few days, because there is usually a progression of organisms, and then over the next couple of days, different organisms will start to grow, others will disappear. Yeah, and this air bubble over here, notice it's also something that I would like to, yeah, you see that those rotifers and some of the ciliates that are there, they will gather around those air bubbles. Because they need oxygen. So that's actually something that you can observe quite often. And maybe over the course of the next couple of minutes, we're actually going to see that this oxygen bubble will grow as more and more oxygen builds up due to photosynthesis. Yeah. Yeah, so that's simply a little bit of a short recap of yeah, last week's water sample, which I'm going to start to follow along also for the next couple of weeks. Okay. So this is yeah, just a little recap, and I would like to now start to show you two short videos of where I collected my other water samples, and I only looked at one of them, so it's going to be also a little bit of a surprise for me, but I was able to find already some larvae here. Yeah. So I'm going to show you now a short video. Okay, just to show you a little bit. Yeah, so this is today in the afternoon. This is basically the place that I usually visit if I quickly need a water sample. It's a fountain, nice Baroque style. And I was really, really disappointed when I went there, because it was so clean. They actually cleaned it up. You see, they're almost, this a couple of years ago when I visited this place, it was overgrown with moss and algae. I found, I don't know, water fleas there, even like a pond. Huh? But nowadays it's really clean, so I was really frustrated at the beginning, because I thought, oh my gosh, I don't know what to show you during the live stream. And then I said, okay, maybe there is something in here. So I lifted up this thing here, and indeed this was actually the place where I was able to collect a small sample. Okay, yeah. So yeah, look at this here. Yeah, so lots of moss on the side. Yeah, lots of filamentous algae. Yeah. And I'm already reading some of the comments here. And that's what I did, is yeah, I simply tried to get some of those filamentous algae off by with a tweezer, which wasn't so easy, because yeah, but after a minute or so I collected a whole tube, and basically here it is. Yeah. Lots of moss as well, so I also collected some of the moss, hoping to find something interesting there. I don't know, we're gonna see. Okay. And then of course I didn't want anyone to fall into this here, so I closed it again, and that's it. Yeah, yeah, that's yeah, we're gonna start again. Yeah, so that's the fountain, way too clean, unfortunately. Yeah, so it's the first place, and I'm gonna show you now another one. Okay, yeah, over here. Yeah, this was in a flower garden, and they've got this little water fountain over here. It's not drinkable, not potable water, and they're using this to water the flowers. And it looked kind of, yeah, that's the flower garden. It was very dry the last couple of weeks, like in many places of Europe. So it's not quite as green and lush as it used to be. But there was plenty of water in here. And what I've done was, with a pipette, I tried to catch some of those larvae. Yeah, because they were very quick. I tried to catch them with my little water container, but they were quickly swimming away. So I was more successful actually by yeah, using a pipette and quickly sucking up some of those larvae. Yeah, and yeah. So Yeah, and that's also scooped up some of the water here. Yeah, so a short question over here. The ponds where I collect my water samples almost never have Senedesmus. Lots of Spirogyra, on the other hand. Could that be of water quality difference? Yes, of course. What happens is that depending on the type of organisms that you find, they can sometimes be so-called indicator organisms. And this basically means that depending on the organisms, this tells you something about the water quality, about the amount of nutrients present, oxygen concentration, and so on. And however, what I've also found is that sometimes when you have to wait a little bit. So what in my case over here, I added a little bit of this wheat grain into the jar, and then after a couple of days, certain algae started to just explode and started to multiply. So this is also a little bit a question of not only of water balance, but maybe even a little bit of luck, which organisms were present in the first place, and if you are able to create the right conditions for them to multiply. What I also sometimes would recommend, also tried that, is to add a very small amount of fertilizer. Mineral so nutrients in other words. So you take a water sample like this and add a tiny drop of let's say artificial fertilizer that you use like for fertilizing flowers. Yeah, this will provide the nitrogen and the phosphorus for the algae growth. Water is of course present, CO2 is present because it gets it over the air, sunlight is present, and then you can also expect a certain algae to grow. Yeah. However, you do not expect that just because you add fertilizer, all of them will grow, because some algae, they don't like certain fertilizers as well. So you see, it's all quite complex, and it depends quite a lot on which organisms you're looking at. Okay, so this is so that's basically the thing here. And now we're going to have a short speaking of water quality. It would be interested to know about how algae affect that. Perhaps different types of algae would mean different things. Could look at some of the samples too for a future video. Yes, thank you. Basically, it's like this: algae will reduce, they will absorb the nutrients, right? And what will happen is that they will therefore accumulate many of the nutrients, and then of course, unfortunately, they also will die off, those algae, and then they will start decomposing, and then they will put those nutrients back into the water, right? So yeah, unless you're removing the algae, the total the amount of nutrients in the water will more or less stay the same. Yeah? So what I've got here, let's have a look at this here. Those 3 tubes we're just going to ignore. I just put them in here. That's you'll never guess. It's an alcohol that's basically artificial meat. So basically you can get those soybean-based meat, which is not real meat. Yeah, so it's called, and I basically preserve this. We can also look at this if maybe in a future live stream. And I've got over here 4 samples. That's the one sample that I collected, and this one over here, that's some moss that I collected from the same place. And over here, this is from this water fountain as well. And in here, there are a couple of larvae, which I would like to have a look at. Okay, and we're just going to take it from there. And honestly, as always, I don't always fully know what to expect, okay? And I just realized that I'm super well prepared because I'm missing half of my equipment. So I need to get myself a couple of Petri dishes. So you have to bear with me for 10 seconds. Here we go. Here we go. I've got a couple of Petri dishes here because it's much easier this way to actually, Let's turn off the lights here as well. And I would say let's have a look at, I don't know, let's have a look at a couple of those, those larvae over here. Okay, I'm just going to show this to you here as well. Let's focus this. Yeah. You can actually see that there are, 3 of them in here. Yeah. There's the 4th one over here maybe. Yeah. Let's have a look at those. And what I would like to do as well is I'm going to later on, I'm going to try to put them back into the tube here, and I hope that after a few days they will actually start to hatch. Yeah, it could be mosquito or related. Okay, I will tell you the following. Strictly speaking, in some countries, I know that in some countries they're really strict about standing water. That's not permitted because it's a breeding ground for mosquitoes, and where you've got mosquitoes, you've got malaria. And as a matter of fact, mosquitoes are considered to be actually the what they call the deadliest animals in the world because of malaria. Yeah, so actually, that's not a good sign to have those. But for our purposes, yeah, we're just going to, yeah, give it a try. In any case, I just realized maybe the thing is still a little bit too large, but I'm going to simply try it in any case. Yeah, some of them are still in here. They're still in here. You know what I'm going to do? Is I'm going to now try the following. I'm just going to go in here. See, this is a huge, I cut off the tip of the pipette. It's too small. So what I'm going to do now is something different. I'm going to take a pipette here, and I have to make it a little bit, I have to make the tip a little bit larger. Otherwise, I'm not going able to, yeah. So I'm going to go like this. And I hope to be able to, here's one. Let's, did I get it? No, I didn't get it. Come on, up. Still not. Maybe it's easier for me to actually take them from here. Because they're... I can't. It's not long enough. That's a pity. So what I'm going to do now is the following. I'm going to add some more water here. Or maybe I'm able to... Let me see. I need glasses. Because I'm already a little bit... My vision is it's much better. So let's have a look. I'm able to catch this one here. See, they're so fast. Nope. Almost. No. Nope. I will try the different one. I'll try this one over here. Yep, here we go. I got it. I need a slide. Where is the slide? I found it over here. Look at this slide here. This is a concave slide. And I'm going to remove all of the excess water. Okay, I think this is going to be fine. I received an email just a few days ago. I dropped something. I don't know what it was. It's just a cap. I received an email a few days ago with a request to do a little bit more stereo microscopy. Okay, so let's do the following. Let's try this using my stereo microscope. And let's have a look at this larva over here under the stereo microscope. I have to take it a little bit. Yeah. And yep, here it is. One of the most dangerous animals in the world. That would actually have been also quite a nice title of the live stream. Not only algae, but we're also looking at the most dangerous animal in the world, which is what appears to be what could be the larva of a mosquito, but doesn't. Yeah, but or related. Yeah. Yes. Yeah. Looks like chironomids. Mm-hmm. Yeah. So the question is now, how does this look like under the as a matter of fact, what I'm going to do, I'm going to put it back anyway into the tube, and then maybe after a couple of days, it's gonna undergo metamorphosis, and then we're gonna have the adult, and then I would like to also mount the adult insect as well. But let's have a look before we look at the algae. Let's have a look at this here. Just a second. Only low magnification possible now. Okay. Where is it? Of course, it's way too quick. Let's do this as way. Here we go. Okay. You see. Here it is. Looks a little bit too dark. Should I try that? I could try to add a cover glass. It's going to squeeze it a little bit, but it is a concave slide. Yeah, so maybe it's not going to crush it completely, but it's also going to limit its movement a little bit. Yeah, so let's try that. Let's try that. It's now over here. I'm going to push it into the center here. No, I accidentally. Here it is. And it's now in the middle. And stop moving. Oh, it's still moving a little bit. Maybe it's sensing that there is very little water. And I'm going to carefully drop a cover glass. Let's turn off the light here on top. Okay. Yep, it did compress it a little bit, but it is in the concave area of the slide. So let's have a look. Here it is. So much better. And then we should also be able to actually look a little bit into the digestive system. Let's add a little bit of color. Yeah. And over here in the back, this is the breathing organ here. So yeah. So I'm just going to, I thought it was Diptera. Well, as a matter of fact, Diptera are those insects that only have one pair of fully developed wings, to which, of course, also the mosquitoes and the flies, they belong to the Dipteras. So it is actually correct. Diptera, basically mosquitoes and related, and also flies belong to the Diptera. Yeah, so one fully developed pair of wings. Of course, we're looking here at the larvae. Yeah, so it is, of course, a Diptera. Yeah. So yeah, so it's this is how it looks like. Let's go up a little bit again with the magnification here. This is already quite dark. And highly pigmented. Look at those fine details. I'm quite sure that these must be some kind of sensory hair, maybe. Yeah. Look at all of the details that you're able to see here. Plus the digestive system. Yeah, maybe we're able to see the peristaltic movement as well. Yeah. Yeah. Yeah, so that's basically, look, to a certain extent, I know that these are not always very nice animals, nice under quotation marks, because they cause so many problems. At least if they are indeed mosquitoes. But in any case, I think under the microscope there is a certain, not only fascination but also a certain degree of beauty under quotation marks. Yeah. So quite interesting, the complexity of nature. Yeah, so that's basically something that I'm going to keep following over the next couple of days, hoping that they will now form an adult. And then the adult insect will be then on the surface of the water, and then I'm able to catch it and hope to be able to make a permanently mounted slide. Yeah. So what I'm going to do now is because I still want to look, of course, at the water samples, I'm going to try to put it back. Okay. And we're going to take it then from there. So here it is. And I have to carefully lift it up again. Here we go. And I'm going to try to put it back into this tube here. All the way down into the tube. They're still alive and happily going. There is another one, appears to be over here. It looks almost like a larva. Another larva, or here as well. And the rest I'm just going to also... I generally like to use the same water and not tap water for the osmotic balance, of course. So, and here we are again, and now they're still happily swimming around. Okay, so I need to, now I need to grab the lid. And and I put it back now into my rack. 3 of them. So I'm quickly going to, composite eyes of a housefly. Yes. So another thing that I would like to do, and over here you have also, that's the slightly larger sample. You can see that there are, what do I give them to eat? Actually, in the past, I've not given them anything to eat, and they still kind of did quite well. Okay, so usually some of the debris that's still in there should be enough. Just a second, I think I have to sneeze. Excuse me. No, I don't. Okay. Yeah. So there's lots of stuff also here on the top. I've also seen, that there is also another one alive in here. The question is now the following. What about this here? Yeah. So I'm going to take this out, and I'm not always very how do you say this? Positive about finding something interesting. But usually if there is something solid, not usually, sometimes if there is something solid, then you're able to get it off. Yeah. But I'm not always very optimistic about that. So we always have to do a little bit of trial and error. Just a second. So, and it's usually, as always, with those pond water samples and so on, it's always a good idea to put something solid, some solid material under the microscope, and to then hope to find something there. But sometimes if the water is, if there's running water, then sometimes this also flushes away any microorganisms that might be present. So lots of debris, but a few interesting things already, even though if there's not a lot of movement all the time. Where is this? There are, I've seen already, there's some, as I said, what are we able to see here? Yeah, it is something sometimes very typical if you put some solid material, because what you have over here is that rotifers, like you've got over here, they like to attach themselves to a surface. Yeah. It looks like the moss is completely soaked in water in the test tube. I'm not sure if it's recommended to do that. Well, no, it's not recommended. But as a matter of fact, this piece of moss was actually floating completely in the water in the fountain. So I kind of scooped it up. Okay, so it's correct that normally moss, should have it very wet, but it's normally not completely submerged. But this one actually was floating in the water, and this is why I collected it, because I was actually kind of hoping to have, to see certain organisms sitting on it, growing on it, and this is actually what we're able to see here, right? There are a whole bunch of rotifers, on this tiny. This is a huge paradise. Unfortunately, the decomposing moss covers up many of them. Yeah. But that is actually something that we can see quite frequently. If you want to see sessile organisms, you have to, of course, put some solid material under the microscope. Yeah. So there's another comment at Microbehunter. For normal use, do you use the video display or the eyepieces more? Trying to decide if added cost of trinoculars is worth it. That's a good one. I'll be very honest with you. I've got a camera connected, right? And the camera, of course, goes into the computer. But for normal microscopy, if I just want to quickly look at things, I always use the eyepieces. And the reason is because you simply, it's a more natural way of interacting somehow, and you simply see better contrast. So the so-called the dynamic range of looking through the eyepiece is simply higher. So you see more details in the dark areas and more details in the bright areas if you look through the eyepieces. Yeah. So for casual use, a quick check, I always use the eyepieces. If I decide to do a video, of course, then I will always turn on the computer and the camera. And I, of course, then document everything with a camera, obviously. So usually I will only do that after I've checked first with the eyepieces that something interesting is present. And when something interesting is indeed present, then I switch on the computer and the camera. Yeah, so that's kind of the system, yeah. Yeah. So simply to show to you here, there's lots of stuff going on here. Okay. Normally, sometimes I think I might have also seen a couple of diatoms as well. Let me check. Ah, here that's a lonely rotifer as well. Yeah, I mean those rotifers are very common. Yeah. Water bears are slightly more difficult to find. Look, this is the other one here at the top. Huh. Here we go. Yeah. So, what I'm going to do now is the following. I would like to show you, before I move on to the next water sample, I simply would like to show you a couple of pictures that some of my viewers sent me, which I would simply also like to share, and I got the permission to share them. So thank you very much. I would like to do this as well. We can, of course, always revisit the water sample. One of the things that I also have, I mean, I've got so many projects going on. Sometimes I'm not following up myself, but one of the things that I would like to do and that I started to do is actually to work on a photographic identification manual for beginners, so that you could basically take a picture or a drawing, and then you have some kind of a reference manual simply to estimate, or not estimate, get a first check of what you're actually looking at. Just also would like to show you something else. I've already showed this to you a couple of weeks ago as well. But yeah, this my next project. If you look at this here, this is a printout of of a diatom here as well. And what I want to do, and I'm practicing, I'm not very good at it yet. I'm practicing of actually now using a pen to actually make drawings of this. Yeah, you can see that this is so what I've done is I've placed this sheet over the other one and then traced it with a pencil, and then try to draw it in such a way. So that is a little bit another project that I'm having right now, is to practice a little bit scientific illustration. Yeah. Okay. I'm just reading your comments here. No, not lacrimaria. This would have been nice. So yeah, Tini Coco almost thought that it was a lacrimaria. Lacrimaria are basically are single-celled organisms with a very long, extensible neck, very fascinating to look at. I have not found so many of those, unfortunately, yet. Yeah. So yeah, but this sample I've just seen is not the most exciting one, because maybe also because of the density is not the highest, of organisms. Yeah. Next sample. So I'm going to put this to the side. Yeah. Unfortunately, I wasn't able to join the Drawing Microorganisms live stream, but I'll need to check it out. Yeah, I wasn't completely drawing it. This would have taken too much time. But a couple of weeks ago, or a couple of months ago, I was actually trying to experiment drawing them. And this is still something that I'm practicing a little bit. But I have to tell you, it takes a lot of time. It's very... it requires a lot of patience. But maybe that's the good thing, that... it gives you a little bit of, requires you to settle down a little bit. So what I'm going to do now is I'm just going to put this sample over here back. Yeah. So this was this moss. Oh, yeah, it was kind of completely floating around, in the water. So next one, I've got, and I know that this one here contains a whole bunch of filamentous algae. So this is one over here. This is also bound to be interesting. So let me quickly. I mean, it's of course also a question. I mean, should I, use those tweezers to go into the other water sample? I would actually be cross-contaminating it. Yeah. On the other hand, excuse me. On the other hand, yeah, so it'll be fine. So I somehow need to grab some of this stuff here. And let's try this and look at this. This is gooey looking stuff. And there is there's simply too much here, so I have to actually really pick it apart and put the rest back. And what we're doing right now is I'm just going to take it apart. Yeah, and what I also plan to do is, I've got so many plans. I would like to also extract the diatoms using hydrogen peroxide. Means if I add hydrogen peroxide to those algae, the hydrogen peroxide is going to digest away everything that's organic, and it will leave the silica cell walls of the diatoms. And this is something that sometimes also looks quite nice. It's a little bit too, yeah. Let's do like let's press it flat a little bit, and let's add a little more water here. So let's give this one here a try. Yep. Yeah, this is actually something that we can see quite often, that water samples where the water is moving or flowing, you often don't see so many moving microorganisms because very often they're being simply flushed away. That's a little bit okay. Let's have a look at a few of those here. Worried about cross-contamination, but still wiping his nose with a tissue he used to for cleaning the tweezers. I will tell you the following, but I'm not so worried. Yeah. But I was cleaning the tweezers first before wiping my nose. The thing is that generally what happens is that those water samples are always in continuous exchange with the environment anyway. Yeah, so look at those here. Yeah, this is actually quite nice looking. And yeah, and there are a whole bunch of diatoms here as well. And if you have a microscope, then you can also do a little, how do you call this, resolution test. There are actually some microscope slides out there that have diatom cell walls, and if you're able to actually see those details here, then this tells you something about the resolution of the microscope objective. Those tiny little patterns here. Yeah, so this one is a very typical case of a natural water sample. You see overlapping algae and so on. It would be kind of nice to have those diatoms concentrated, but this would actually mean that I'd probably have to centrifuge them or to digest it with hydrogen peroxide to remove all of the organic material. Yeah, some of them are actually moving as well. Yeah. So this would actually be then, one of those projects where you simply take pictures of the different types of diatoms and then have some kind of maybe a photographic reference guide with or without drawing. But let's go down with the magnification again. And let's have a look. Yeah. So, yeah, this is the one from before again. Okay. What I've also seen occasionally is that after a couple of minutes, the diatoms will actually start to spread out because some of them actually move in a gliding way, and then they start to spread out a little bit over the microscope slide. So let's go down here again. Yeah, but again over here, no rotifers, no ciliates, at least not that I'm able to see it. Yeah. And yeah. So what here's one in the center tumbling around. Yeah, but again, always in moving water, it's always a little bit of a problem to find those organisms, and therefore it's better to dig a little bit into the sediment of a pond, for example, where there is less movement and you find more ciliates and flagellates and so on. You find them more there. I'm occasionally reading the comments. So what I'm going to do now is I'm going to now show you a couple of pictures. Okay, that somebody sent to me. Okay, and let me quickly check. Okay, I got a little bit. I got a picture. Yeah, where is this? Here it is. Okay, I got here some. I want to quickly show this to you. So first of all, thank you for showing this to me. This is a microscope modification, and I kind of like this as well because, let me start at the very beginning, because this is actually taken through, so this shows the surface of a leaf, okay? Here are a couple of slides. And if you've joined the live stream, then maybe you can also comment on here. This is a microscope modification. So what you've got over here is the following. I would like to this looks like almost like a very basic microscope, but the quality is surprisingly good actually, because there were two modifications made, and I would like to quickly just press pause here and show it to you. So thank you very much to Sasiri who sent me the pictures. Okay, and here's the arrow, and this is of course the microscope, and modification number one is look at the eyepiece. Okay, this is a significantly better eyepiece. It's a standard 18 millimeter field of view eyepiece, and this has improved the image quality significantly, even though it's not one of the most advanced microscopes, but it kind of shows already that just by exchanging the eyepiece, you can actually improve it quite a bit. And that's modification number one. And modification number two is, where is this? Modification number two is, so that's how it looks like. It looks like onion cells. Yeah. Surface of a leaf. Yeah. Modification number two is this one down here. Oh, gee. I have to find it. Here we go. See, it doesn't. Why does it not do that? Yeah, this is the one over here. This is basically a homemade condenser. So this is seems to be a lens that was glued in here to focus the light better. So it's like a condenser which also improved the contrast. So and also the image. So those two improvements, those two basic improvements, actually allowed for the making of significantly better images. So it kind of shows that also those basic microscopes, are able to produce surprisingly good qualities. That's actually the reason why I wanted to show it to you, and that's why I'm quite happy that this one is a little small, but of course you can see that over here on the side there is it's a little bit blurry because it's not a plan objective. I mean, but that's fine. We're talking about a very surprisingly good quality, considering the fact that it's not such an expensive microscope. So basically, take-home message, also something that I did not expect: exchange the eyepieces. Get yourself a better eyepiece, and yeah, yeah, exactly. It's I got a similar one also when I was a kid. Seriously, long time ago. Yeah, almost looks like a toy microscope. Yeah, but yeah, here over here, this is the yeah. It seems some kind of a lens element that was used to kind of concentrate the light more, and then of course also the improved eyepiece. Yeah. Just wanted to share this with you because I kind of found that to be quite nice. Yeah, I would like to show you something else as well. Yeah, also, I think Mike just sent me before the start of the live stream. Isn't this also a beautiful diatom here? Okay, so if you've joined the live stream, maybe you can also write in the comments a little bit. Okay, so thank you very much as well. Just a couple of minutes before I started the live stream, I downloaded those pictures and I also got the permission to share those. So yeah, very nice looking. And that's actually what I wanted to do as well, is if I'm able to or somehow make a collection of those pictures, then we'd have a slight, a nice identification book. Yeah. Is this focus stacked? I'll quickly check the email. Just a second. Let me quickly check. Yeah, I can read the email from Mike. I believe the attached image is a freshwater diatom from a local pond here in Connecticut in the United States. I find lots of them in my water samples, but the one looks exceptionally interesting. Yes. And no, I don't know if it's focused stacked or not. And the second image I want to show you is some kind of a worm. It's from the same pond that the diatom is from. I tried using 3 different AI services to identify it, and I got 3 different answers. Yeah, so that's also something that I've tried to do, is you copy paste the picture. Here's the worm, the screen capture. You copy paste the picture into an AI system, and you ask it to identify it. And yeah, Mike is in the chat. Thank you very much. Yes, it's a stacked image of a diatom. Okay. Thank you very much. 25 stacked images. Yeah. So that's here it is again. I mean, it's just great. I mean, really, it's unbelievable what you're able to do these days. Yeah, with a little bit of imaging technology. Yeah, looks really nice. Yeah. So, and yeah, would be I know, for example, for making if you use, I don't know, many of you, some of you might be using the app iNaturalist, where you can upload pictures as well, which can actually also serve as an identification platform. Yeah. And this one over here, I was kind of wondering a little bit when I looked at that. Where's the arrow again? I was kind of wondering a little bit if, and I don't know, but I've seen this also sometimes. If you look over here, it looks almost as if the worm is dividing a little bit. Okay. This could be, I wonder if it's normally my first instinct, it's an oligoheat worm, some kind of a worm that has bristles on the side, but I could not see any bristles, right? But then I saw this part over here and I was wondering if this could be maybe the mouth, if this could be maybe some kind of a flatworm and not a segmented oligoheat worm. Okay, just guessing. Yeah, because many worms, segmented worms, they have bristles on the side. Yeah, but this one, yeah, and segments, but this one does not seem to have those. But there seems to be this weird opening here, and this could be maybe the mouth. Yeah, I'm not quite sure. Yeah, so I'm just saying. So yeah, so maybe a flatworm. I don't know. Yeah, but in any case, quite interesting. Yeah, because oligoheat worms, they have segments and they have some bristles and so on, or polyheat worms. But this one does not seem to have those. Yes, it does have bristles. Okay, yeah, maybe you just don't see it here because it's a screenshot. Yeah. So yeah, but these are things that yeah. But then on the other hand, there are so many different organisms out there in nature that sometimes probably you'd have to do a DNA test. Yeah, yeah. Would you re-show us your favorite slide? I'd have to. You know what? I'll do the following. Marie Chardon asks me, would you re-show us your favorite slide? I'd have to grab them, but you know what I'll do is I'll write this down and I'll maybe make a collection of my favorite permanently mounted microscope slides, and then I'll show them to you next week. How's that? Okay, so let's move on again a little bit. Yeah, so that's the one. And now this is, there's another water sample, and this is basically the moss. Okay, so I collected some moss from the side, and I don't know if this one actually is that revealing or not. We're just gonna try this, okay? We're gonna just try this as well. So what I'm gonna do is I'm gonna take some of this stuff out here. Desk view again. So there's another interesting phenomenon. The interesting thing is that more people seem to ultimately watch those live streams than my other videos that I made in the channel. Look at this, okay? And over here, down here, there is lots of solid material. So I'm gonna directly have a look at this first. So now I'm going to take a new tip. So I'll be honest with you. Actually, I wanted to go into the forest. You have not seen this. I wanted to go into the forest to collect some moss, with the hope of maybe finding again a couple of water bears, tardigrades. But it's been so dry in the last couple of weeks that I thought maybe it's not going to be very successful. So I decided to therefore grab a couple of water samples again, especially when the temperature is so high as it's been, and usually this favors the growth of algae. Okay, and let's have a look at this one now. You know what, I'm just going to leave the other slide in here. So that's the old one. So let's have a look now over here. But I already see that this does not seem to be a very interesting sample. Oh, but it's also something where we learn something. If a sample looks like this, then normally what I usually do is I do a time lapse, because usually there is more life present than what you would assume, but you just don't see it because the movement is too slow. Yeah, I mean, occasionally, but this is indeed a more on the disappointing. We could use this opportunity to have a dark field view. That's kind of interesting. Why am I not getting a dark field? Look at this. I know why. I forgot the condenser. So that's the dark field view now at 4 times. Yeah, it doesn't look a lot different. So this basically means I'm going to have a look if I'm actually able to see something at a slightly higher magnification. But this is not the most interesting specimen. I mean, you of course can still see that there are a couple of diatoms, plant debris, but it's not the most interesting one. Something is moving here. Whatever that is. Yeah. I think the algae from before were actually more interesting. Again, those fan-shaped diatoms that we've seen before. So, but what I'm going to try now is something different. I'm going to now do the following, and I don't know if this actually works or not. I'm going to add a little bit of water with the desk view. And try to rinse out whatever is in here. but because we've not seen a lot before, I'm not very confident here either. But just put the rest back. And something like this. So, and then usually wait a little bit. I mean, there is movement going on in here. I can see that. Let everything settle down for a minute or so. And then we're going to give it another try. And then I'm going to show you, also checking the time a little bit, one of my failed experiments that I've tried yesterday. But there is movement going on. I wonder what that is. No idea. I'm just checking. Still some stuff here. I'm just going to try it now without a cover glass because there seems to be quite a little bit of sediment in here as well. I don't want to crush whatever it is. So let's try this. So, and yeah, there's something is moving. Just have to find it. Ah, yes, that's a worm. Okay, this is a pretty huge one. How many worms are there? Yeah, that's a worm. Okay. Actually, there are two worms here. Yep. Okay. Yeah. So over here we definitely see those bristles. Yeah. Yeah. So quite interesting here. Moving again way too fast. But okay, these are easy. That's an easy specimen. And what I would like to do now is maybe there is some other stuff here. Yeah. As well. Let's just quickly give it a scan. If there's anything else of interest. Uh-huh. What's that? What is this? Is this? Let's see. I'm hopeful. I am hopeful. I'm hopeful. Is it a rotifer or is it a water bear? This looks very much like a water bear. Yup. Could it be? Yes. Ha ta ta. Tardigrade. Here we go. Yeah, they like to cling to solid material. Yes, it's a tardigrade. Okay, at least at least we were successful. That's a little bit for for people who are starting off with microscopy, kind of finding a tardigrade or your first tardigrade. It's almost like a rite of passage, you know. It's yeah, a rite of passage basically. Once you've managed to find those, then yeah, yeah, they're called moss piglets as well, and because they're commonly found in moss. And they are, of course, surprisingly dry resistant. So that means they can actually survive all the periods of dryness. But this one over here, because the moss was always wet, yeah, kind of happy that, yeah, getting it separated now. So normally what I would do is I would like to then usually take a pipette, try to suck it up, and then put it on a separate slide. This takes a lot of patience. I mean, I could try to do that, but this is a little bit risky. And I don't know if I'm going to be very successful with it. There's always the danger of actually losing the specimen this way. So what I'm going to do is I'm going to add a little bit of water here. So I would like to separate this tardigrade from... this is going to be super challenging. This is going to be extremely challenging. I don't know. I'm going to... You know what I'm going to try to do? I'm going to try to risk it even if I might lose it, okay? So maybe I'm going to lose the tardigrade, okay? It's quite likely. It's now right in the center here. It's right over here. So what I'm going to try to do is I'm going to try to pick it up with this pipette. And this is going to be really difficult. I hope I was able to suck it up. And I don't know. We're going to see if I actually managed to transfer it over here on this slide. If not, then also fine. You see how much other debris I was able to take up. But maybe I did not catch it. Or maybe I did. Actually, I did. So this now is good news. You see, it still likes to hold on to debris. So I'm going to try now the following. I'm going to now add a cover glass. And then I'm going to try to find it again. This is going to be a super big challenge. Cover glass added. So it's got to be... Again, patience is needed. I don't know, is this the one over here? Yeah, if we don't find it. So this is normally the time when I would look directly through the microscope eyepiece. Oh, we were lucky. Look, this actually was faster than anticipated. Yeah, and because I have a cover glass now connected, whatever term, over it, I can actually go up with the magnification. And 40 times. So here we go. Yep, so that's basically a little surprise. That's the tardigrade. Yeah. So I'm going to quickly, DIC. Yes, I know many of you say how would it look like in regular? That's regular bright field. You see how much more light there is. Bright field has the advantage that you're actually seeing also the real colors. Yeah. It struggles to get off the moss. Strictly, I don't think that it's actually struggling to get off. They very frequently cling to the moss with their hind legs. So yeah, they actually don't want to let go. Yeah, so sometimes I actually had to take again a pipette and try to separate it to simply completely isolate it. And then as soon as it finds again something to cling to, it will attach itself to that. Yeah. So I would probably say it's not struggling to get off. It doesn't want to get off because it's simply, yeah. I think maybe it's looking for something else to hold on to. And because we're right at it, quickly, how does it look like with this is now phase contrast 40 times. Yeah. But sometimes we don't even see it that well. Yeah. Yeah, so I think we're just going to, yeah, just probably just leave the other one. Yeah. Yeah, so this is a little bit. Yeah, so actually, in that sense, yeah, I'm also quite relieved that I was able to show you a tardigrade today. Not only moss, but also some micro animals. I know that I've said this already many times before, but one of the things that fascinates me about those animals is that they're so-called eutelic. This means that the number of cells that they have is fixed. So adults of these animals, they have a fixed number of cells. Yeah, and this means that every individual cell of those animals can be traced back to the first cell. So this is very different than we humans, for example, because different humans have a different number of cells, obviously, right? But with those simpler animals, they don't show that. They actually have all of them have the same number of cells when they're adults. It's kind of interesting how scientists actually found that out. I had to do a little bit of research because the same is the case with rotifers and some nematode worms. How do they actually know if that they have the same number of cells? And what they have done is they've actually done some staining and they counted the nuclei and they discovered that the nuclei they have the same number of nuclei. Yeah. So this means that yeah, some time ago they actually did some cell counting. Yeah. Does it have a brain? That's a good one. Why is it a good one? I made a video about this a couple of months ago. No, they do not have a fully developed brain. They have so-called ganglia, and ganglia are basically a collection of nerve cells. So it is not a fully developed brain like we know it. However, it's not completely decentralized either. So there are actually a collection of nerve cells that kind of function a little bit like a brain. Of course, a higher thinking ability or intelligence is not something you don't have here. Yeah. Would you consider it conscious? And that's another interesting one, honestly. And I want to make videos about this because this is actually something that I'm asking myself as well. And I would say now the following, and I'm making a claim now, and I know it's debatable, okay? I claim that those simple animals do not have a form of consciousness because they do not have a brain. To the best of our knowledge, you need a complex brain to have something like consciousness or self-awareness or generally awareness. And this is almost a philosophical question, but if you assume that you need a brain for consciousness, then those animals probably are not conscious. Now, some people are going to say, and I understand and agree, well, but they do seem to behave, under quotation marks, they do seem to behave intelligently. Whatever that means, right? For example, it's trying to move forward, it's trying to find food, it's trying to, avoid certain unfavorable conditions, it moves towards oxygen. All of these behaviors are responses to the environment and they kind of seem to make sense, right? However, you have to understand that those responses are like reflexes, and you find also responses like this in other living things, in single-celled organisms. Paramecia, for example, they move towards oxygen. Chlamydomonas and other algae, they move towards light. So the response to environmental stimuli is almost separated. Okay. What does it say? Reconnection successful? Are you still? I hope you're still able to see me. I don't know why it made a. I don't know why it made a short message. I hope that I'm still online. Okay. Yeah. Okay, good. The computer always all of a sudden gave me a short message, reconnection successful. So why reconnection? There shouldn't have been any. Okay. You paused. I don't know why. Okay, but it reconnected automatically. That's the good news. Yeah. Yeah, I was talking a little bit, almost like a water is antenna for consciousness. Yeah, let's put it this way. What is consciousness? You see, that's very difficult. Even, yeah, good. So there was some kind of a short break in the transmission, first time ever, but apparently we're back to normal. I'm going to try something else. This is going to be risky because the working distance is too small. I'm going to go up with the magnification yet further, and I might touch the cover glass or not. No, I didn't. Very good. So that is now the 60 times objective. Okay. Yeah. So another thing that I want to say is the following, because I was talking about cells. Back in the day, in the 19th century, when scientists formulated the cell theory, which states that all living things are made of cells, plants and animals. The cell theory was first defined for plant cells. All plants are made of cells. It was Matthias Schleiden, a German biologist, came up with that statement. All plants are made of cells. And at that time, this was easy because the cell wall was so visible. But then later, with Theodor Schwann, another scientist, he said, well, all animals are made of cells. But now, where can you see the cells over here? A tardigrade water bear is made of approximately 1,000 cells. But honestly, it's almost, I cannot see them because those animals, they do not have a cell wall, right? So we do not really see where one cell stops and the other cell starts. Yeah, so simply to realize that even animals are made of cells was quite remarkable. I'll tell you why from a philosophical perspective. Because it actually means what? Plants are made of cells and animals are made of cells? Really? Does this mean that plants and animals and all living things are somehow similar in that sense? Because we're all made of cells, right? So this was already a huge discovery at that time to actually realize that on a very deep biological level, we humans or animals are not fundamentally different from other living things like plants, because we're all made of cells. So this was, I think, the discovery of the cell theory back in the 19th century also put the position of humans and of other living things a little bit into new perspective to kind of show that all living things in that sense are share certain similarities. Yeah. So, consciousness is a human idea. It does not have a yes or no answer for the real world. Yeah, that's a way of seeing it. It's, I would say maybe psychologists would say that consciousness maybe is a social construct, I don't know. Yeah, it's very philosophical. Yeah. There's still more questions than answers when it comes to microbes. Yep. The more we know, the more questions appear. And for me, it's fascinating how, and one of the reasons, how do we, just another thing, when we look at this creature over here, sorry if I'm getting a little philosophical now again, we immediately realize that it's alive, right? Everyone would agree, yeah, it's alive. But how do you know? And the reason is because it moves, right? But there are many things in nature that moves. But the reason why we consider this alive, and let's say not an engine, spinning and rotating, is because the movement is to a certain extent unpredictable, and it appears to be a little bit random. Yeah, a spinning engine, we know it's not alive because it's always a repetitive motion, right? So it can't be alive. But if we look at a tardigrade like this over here, I don't know into which direction it's going to move in the next minute. I cannot predict it. And I think that this unpredictability kind of also hints a little bit towards living systems. Yeah. So, yeah. No, there are pigmented tardigrades, sure. And if you see, for example, in a very basic one over here, if you look into the body, you're going to see that it's mostly transparent, but the inside of this tardigrade seems to be slightly darker, and usually the digestive system takes on the color of the food that it eats. So yeah, if it eats a lot of algae, for example, it turns more green. However, some tardigrades actually have a pigmented pattern as well. And that pigment is also used for identification. Yeah, it thinks about survival, to eat, to drink. All living things, of course, need to survive. But the question, of course, is does it do that consciously? Yeah, and I probably don't think so. Yeah. Yeah. If you build a machine that acts like a tardigrade, would you consider it alive? Honestly, that's a good point, and I will. I want to challenge you with a question that I also ask my students. I mean, and maybe I've already mentioned this in this live stream. My wristwatch, right? Here, it's a little bit of retro looking from the 1980s Casio watch, right? It moves like a living thing. It consumes energy like a living thing. It responds to the environment. When I push a button, it responds like a living thing, right? And to what extent is my wristwatch now alive? That's a little bit the question, right? And some scientists are actually saying, or philosophers are saying, maybe the distinction between alive and not alive, it's not a clear-cut line. Maybe there is some kind of a gradient, that certain things are supposed to be considered more alive than others. Now, I know this is kind of getting carried away a little bit, right? But the reason why this is of interest is especially when, for example, in space, you're looking for living things in space. What are you actually looking for? Right? How do you actually know if you find some interesting stuff moving around on another planet? How do you actually want to know whether it's alive or not? Yeah. And to make matters worse, philosopher once told me, actually a biologist who was into philosophy once told me, well, actually you cannot even define life. Because if you were to define life, then you would define who you are yourself, and that would be going in a circle, and that's philosophically illegal. You cannot define yourself. Yeah. So life cannot be defined; it can only be described. Yeah. This is a question. The wristwatch does not reproduce, right? So reproduction, however, is an essential, characteristic of being alive. But what happens if you now have a living thing that does not reproduce? I want to give you an example: red blood cells. Red blood cells, don't have DNA. They are cells, but they don't reproduce. Are red blood cells alive? And some people consider, no, they're not alive because they do not have the ability to reproduce. Yeah. So there are conditions for life. Yes, there are some characteristics. Yeah, and one of the most important characteristics is the presence of a metabolism, so that they basically eat food, they metabolize it, they do chemical reactions, they produce wastes. And if you have metabolism as an important criterion, then you have to ask, as the next question is, how complex does a chemical reaction have to be for something to be considered alive? So you could imagine in space, in the universe somewhere, a cloud floating around, with complex chemical reactions happening. Is that cloud alive? It's not my idea, right? But it's it kind of is a mind experiment to kind of stretch a little bit, or to explore a little bit. What does it even mean when we're looking at something that's alive? Is in the cloud that's kind of moving around and changing shape and doing chemical reactions? Is that alive? Yeah. So so these are questions a little bit. I know I was drifting off a little bit into philosophy here as well, but these are indeed questions that I ask myself. And one of the, I forgot his name, this German scientist who first has discovered tardigrades in the 1700s. I read his original publication. I actually found the scan online. He made a drawing of those. He actually called it the little water bear. And he basically realized something, and this is one sentence, really interesting. And he actually saw that one of those tardigrades under his microscope got injured. He saw a little cut there, and it died under the microscope, right? Nothing unusual, right? So we see this happening every time. But the comment was very interesting that he wrote. He basically said is he found it fascinating that even microscopic animals like these are able to show or display suffering and death. Yeah, so he kind of realized that even microscopic animals are able to show those characteristics that we're actually able to see also in larger animals. So this was again a big realization that, strictly speaking, even larger animals, maybe even we humans, on a very biological level, not that different from those microscopic animals. Yeah. Yeah. Euglena, very nice example, Marie Chardon. Here's like Euglena between plant and animal. Yeah. So this is the thing where scientists at that time classified, yeah, if the organisms do photosynthesis, it's a plant, right? If they're heterotrophs, if they eat other food, it's an animal, right? And then all of a sudden you have Euglena, which is a single-celled organism, which is able to do both. It's able to eat food and it's able to do photosynthesis. Big confusion. Is it now a plant or an animal, right? And yeah, it's neither. It's a protist, a protozoan. Yeah. So this kind of also showed a little bit the classification systems that people used at that time. Nature does not want to be always put into boxes. Yeah. Yeah, so I got a little bit carried away here. Yeah. Another comment here, there's also evolution, which seems to be a very important part as well. Yeah. Yes. Yeah. Yeah, those are the behavior of certain organisms. If we've always traditionally it's always been said is if an animal is strong enough, then it has a higher chance of surviving, right? If an animal is resistant against certain diseases, it has a higher chance of surviving. But actually, evolution is not only on physical characteristics, but also on the ability to learn, for example, and also on behavior. Yeah. If a certain behavior increases the chance of an organism surviving, then they also have a higher chance of passing on their DNA to the next generation. Yeah. Yeah, so yeah, it says here, a little philosophical dialogue is now and then is a welcome tangent. I tell you why this philosophy is a little bit interesting as well, is because when I do microscopy with my students, for example, insects, catching insects and putting them under the microscope, many students actually do have ethical concerns. Is it okay actually to kill an insect to observe it under the microscope? You have to kill the insects because otherwise they fly away. Or what happens if I now basically, I don't know, just leave the slide here under my microscope and it's going to dry up and then the microorganisms and animals, they die? Is this moral, so to say, you know? That's a very common question that people students also ask me, and I also got in the forum and in the chat, people ask me this. Yeah, and the answer that I have is well, is it? I'm challenging now. Yeah, is it moral to scratch my skin when it itches? Because what happens is I'm killing off a lot of skin cells as well. Right. So, and the thing that I want to point towards is that this concept of individuality, as we know it for higher organisms or humans, sometimes don't apply to single-celled organisms or microanimals. Yeah. So, yeah, and it's for science, sure. Yeah. And then the other question is, of course, is if students don't learn to respect and appreciate nature or love nature for what it is. Yeah, you can only do that if you maybe get a personal connection to it, and so maybe, yeah. I have to tell you another story, and then I'm gonna quit with philosophy. Okay. Actually, I wanted to show you some algae, but we ended up talking about philosophy with a tardigrade here. Yeah, how many organisms do you kill when you walk through grass? Correct. If I eat food, if I swallow yogurt, I'm billions of bacteria. There was actually a couple of years ago, this was in a zoo here in Austria. It was, for those of you, it's in the Viennese Zoo, which is one of the oldest zoos. And I remember that it was a couple of years ago, it was big in the media. I think the director decided they had to kill a giraffe. And they wanted to feed it of course, they're going to feed it to animals, and they had to do that because there was a biological reason behind that. Yeah, I don't know if the animal was ill or whatever, but in any case, it was necessary and people got really upset about that, understandably. And then he said the following is I'm not so much concerned about the fate of the individual animal. What's more important is the population. So it's not so much whether this individual tardigrade survives or not. What is important is the biodiversity, that this is maintained. Yeah. So it's never about the survival of the individual. It's always about the survival of the species. And that is a connection that some people don't get. Right? Yeah, because people get emotionally attached to an individual organism, to an individual animal, and not towards a whole population. But biologically, what is important is that the whole population survives. Yeah. So you see that this is an interesting disconnect between the way that the scientist thinks and how obviously the general population thinks. Yeah. So as a scientist, okay, just I don't know, add some alcohol here. Let's kill the tardigrade. Who cares? Because as long as the biodiversity of the tardigrades is not endangered, yeah. But yeah, but from an individual perspective, this is something that people don't like. Yeah. So you see that this is interesting disconnect here. Yeah. Yeah. Every time I heat water from a cup of tea, I'm killing countless millions of bacteria. Sure. Yeah. When I swallow something, yeah, I don't know. Every time when we eat something, what was there? Every person eats a couple of 100 grams of insects every year. When we eat, because we're in the food, yeah, there are so many insects that cannot be removed in bread and so on. Yeah, and if a leech attaches itself on your skin, are you going to leave it there? I want to get it gone, right? When a mosquito sucks my blood, what am I gonna do? Of course, I'm gonna, yeah, yeah. I have no problems killing mosquitoes, right? Yeah, mosquito larvae, they're bad, obviously, right? Or they do carry diseases, right? But those cute little tardigrades, dang, yeah, don't want to kill those. Yeah, but strictly speaking, it's a question of it's of emotional attachment. Yeah. Okay, I really got sidetracked, but sometimes, yeah, those philosophical excursions are also quite, at least to me, interesting. So what I'm going to do next week is the following. Yeah, so we looked at the 4 water samples. We got stuck with the tardigrade for way too long. Hope you found it interesting anyway. No, I do wanted to show you something, but maybe by next week I'm able to. I promise this to you. So just for a very quick failed experiment. Okay, I'm just going to show this to you as well. Desk view. Let's put this away again here. Failed experiment. Work in progress. What I have here is I kind of put this a little bit. I found a post on Reddit, and what I've got here is this is plastic. Okay, it's plastic. Okay, so and what I've done is I've drilled a hole in here, and I pulled some hair through the hole using this little device here. If I've got it, you see it's used for threading needles. Where did I put it? Here it is. Oh, they sent me a whole bag. I don't know if you know these things here, right? It's for threading needles. So what I've done is actually I pushed it through one of those holes here, right? Yeah, like this, and then put some hair in here. Yeah, and then I pulled it through to get the hair in, and I fixed the hair with tape on one side, and the hair was standing out on the other side, and I added some nail polish here to glue it in place. And then I used a sharp razor blade to cut it off. With the idea is I wanted to look at the hair perpendicularly, right? It didn't work. Okay, so this is a little bit, a little bit the thing. So I'm just going to quickly show this to you. It's a little bit too dark, but I wanted to actually have a look at those hair right from the edge on, and that's actually the last thing I want to show you. And then live stream today has been a little bit longer anyway than I intended. Yeah, but it's too dark, so unfortunately, it didn't quite work out the way I wanted it to work. Where is this? Nope, that's the other. I have to find it. Just a second. Yeah. So I'm just going to show this to you why it did not work the way I hoped it to work. Because the hair is too dark. So sometimes failed experiments are also quite useful. So I'm gonna turn on the arrow now. So that's a little bit now my mouse is so here. Do you see those round things? Ah, my mouse is not. Do you see those round things here? Right. That's basically the hair seen from the top. Yeah, here, here, and you see that they're way too dark. I was kind of hoping to see the canal. But it's unfortunately too dark to see that. But that would have been actually a technique. I'm gonna go up with the brightness. That would have been a little bit a technique to actually look at it. Perpendicular top illumination. I tried using my stereo microscope, and the problem with the stereo microscope is that it is too small. Okay, so that is a little bit the problem that I have, and I don't have a very strong flashlight with me. But that would be something where I'm trying to work on. I want to make basically perpendicular cuts, transverse cuts, so to say, through very thin objects. Yeah, it's too small with the stereo. I can show you that with the stereo. Last thing to show you today. Where is it? That's this one over here. Yeah. So you actually see the arrow. Yeah, so that was the hole filled up with nail polish, and here you see the hair. Yeah, so this is kind of, it's all a little bit pressed to the side. So these are the hair, and let's go up. Yeah, and I have to refocus again a little bit. And here you might be able to see a little bit that some of the hair are a little bit brighter in the center. Yeah, so this could be, the central bright part, but you see, not very, not good enough. Find exactly. This is actually, there is a technique also in Reddit. I've seen that somebody actually tried to do that and actually got way better results. It could also be a little bit that the hair maybe is a little bit too dark. Yeah. So just sharing this with you. It looks hollow. Yeah, the central part is the hair is not hollow, but there is this central part which has a different color, right? So this is something that I'm kind of working on a little bit. I just wanted to share that with you. Not fully successful, but work in progress. So what I'm gonna do now is I think might be worthwhile cementing and thin section it in mineralogy. Yeah, in resin first, thinking of maybe embedding the hair in maybe some kind of a matrix, maybe epoxy, maybe. Yeah, I think there's plenty of experimentation here, right? So what I'm gonna do, folks, is I'm going to call it a day today. I've done it a little bit longer anyway than I intended, but I hope you found it interesting. Also a little philosophical excursion, which I did not plan to do. But I wish you all the best and hope to see you again in an upcoming live stream. Bye-bye, and thank you for joining.